According to the law of Mendel, only one gene gets expressed. One allele, if it is the unmodified or the modified allele, they may start to become dominant. According to the law of segregation, here the genes will not get expressed. Hello everyone, a warm welcome to another session on the principle of inheritance and variations. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had studied about the way Mendel conducted various experiments in order to bring about cross-pollination, so that in the coming progeny, what kind of traits the progeny or the character the progeny will carry forward can be understood. For that, he used various binomial expressions, that is, he used a mathematical formula, and also he applied or used Punnett square. And we also studied about the monohybrid crosses and about back cross and test cross. So in the previous session, we studied only about his experiments. So based on those experiments, he actually put forth two important laws so that he can explain it to the people. So what are those two laws? And also there is one more inheritance which is not put forth by Mendel, which is called as non-Mendelian inheritance. So what happens in that non-Mendelian inheritance, which is also called as incomplete dominance or codominance, we shall see. So moving on to the session, we shall first study about Mendels Principles or laws of inheritance. So, in order to make the people or other geneticists or scientists understand what his theories were based on the experiments he conducted, he put forth two important laws which are called as Mendels Principles or Mendels Law of Inheritance. So, the first law, it is called Law of Dominance, and the second one is the law of segregation. So, we shall study the first law of dominance, wherein he said that, remember that genes are present in a plant. So, earlier they were called as factors. So, characters or traits in a progeny are controlled by discrete units called as factors. So, these factors were later on called as genes. So, these factors or genes, they usually occur in pairs. So, we had already seen in the previous session, they usually occur in pairs, either this or like this. It will not get expressed single. It will not get expressed single like this. This is wrong. They usually occur in pairs. So, Mendel, in his first law of dominance, he put forth that the characters of the progeny are controlled, or the characters of an organism, in his case the pea plant, were controlled by certain units which he termed it as factors, and later on he gave them the name genes. And he said that these factors or genes, they usually occur in pairs. They never express singly. They usually appear in pairs. And he also said that if the pair is a dissimilar, so this is a dissimilar pair. So these two are similar pairs. This is a dissimilar pair. Why dissimilar? It is heterozygous. Why? Because it has a dominant gene responsible for tallness, in this case, in the example which I have taken here, and the recessive gene that is responsible for the dwarfness or the short plant. So, if the pair of factor or gene is dissimilar, then one member of the pair, that is one member of the pair will dominate and the other will be recessive. So, this member will be dominating. So, this member will be dominating and the other will be recessive. So, this is what he explained in the monohybrid cross, right? So, we'll just briefly brush up with what we had studied. So, what he did was he made a cross between tall plant and the dwarf plant in the case of monohybrid cross. So here, to explain his law, what he said was the characters are controlled by factors. So what are the factors here? The two factors which have considered here is T and TT. This is responsible for tall. So this factor brings about or expresses tallness in a plant and this factor expresses dwarfness in a plant. And what did Mendel say according to his law? They can never occur in single. They are always in pairs. That is why we write two alleles here or two factors here. The factors always occurs in pair. So what he did when he made a cross, by putting the Punnett square, we can find that so he said in dissimilar factors, one will be dominant. So can you see here in the dissimilar factor, one is dominant and the other is recessive. And which factor will get expressed here in the plant? The dominant factor. Therefore, this plant will be tall. Again here, one is dominant, the other will be recessive in the pair of factors, in the pair of allele or in a pair of gene, that we say one will be dominant, the other will be recessive. Again here, the dominant will suppress the expression of the recessive character, it will be tall. Again here, it will suppress the expression of the recessive character, it will be tall. Again here, the dominant will suppress the expression of the recessive character, they will be tall. So that is what he meant in law of dominance. So according to his law of dominance, in dissimilar pair of factors, one member will have, that is one factor will be dominant and the other factor will be recessive, and always the dominant will get expressed and the recessive will be suppressed. So that is what is meant by in his first law of inheritance, which is called as law of dominance. So next, moving on to the second law, that is called as law of segregation. So how did Mendel put forth these two laws? It is based on his experiments that he conducted, which you had studied in the previous session. So here in the second law, which is also called as law of segregation. Segregation. Say for example, you have a set of books. That is, you have 5 biology books and 6 physics books. So now it is mixed up. Now you're separating them. You make biology one bundle, you make physics one bundle. You're separating the character. What is the character of the textbook there? The one of the textbook is biology, that is the character of the textbook. The other textbook is physics, that is the character of the textbook. Those characters are mixed up. They are jumbled now. Now you're separating the characters, wherein you'll make a set of biology book and you'll separate a set of physics books, right? So similarly here, law of segregation means you're segregating the characters. Here, during the gamete formation, so gametes, they will have a male gamete and a female gamete, wherein the male will always be, the trait will be dominant and the female will be recessive. So during the gamete formation, the factors or the alleles that are called, they get segregated in the progeny. So this best example which he explained for this law of segregation, which you studied in the previous session, was the crossing that he did between the violet flower of the pea plant and the white flower of the pea plant. So, did the violet and the white flower mix together and did a pink flower get formed there? No, right? The violet flowers were expressed separately, the white flowers were expressed separately. So, that is nothing but the law of segregation, wherein the genes get segregated. So, to explain, again we'll go back to the previous session what you had studied, wherein he crossed between the violet flower and the white flower. So, a cross was made. So, what did he get when he put a Punnett square? We shall see. So, here W, capital W, the recessive one small w's, capital W, capital W, capital W, capital W, small w. So all the flowers were violet. Now, what I want you to understand in this law of segregation is, though here white flower was there, did it mix with the dominant character and did it become a pink flower? Did they combine? No. They got separately expressed as violet flower itself. Why? Because violet was dominant over the white. This is nothing but law of segregation. So, this is what happens in law of segregation. They get expressed separately. They will not mix. So, if I write here, okay, the gene having a violet flower and the gene that is responsible for the white flower, both are there. So, if they mix, you might have seen the color palette while if you love painting. So, if you mix violet and white, maybe you will get a pink color, right? So, you might think, okay, now the genes, both the genes have mixed. They should be a pink color. No. According to Mendel, here the genes will get segregated. That is nothing but the law of segregation. Here, in spite of that, the violet only will be expressed. Violet only will get expressed. Both of these will not combine with each other and give a pink color flower. It is not a pink color flower. Pink color flower will not be formed. Only violet will get expressed. So, if it is a recessive gene, then only white will get expressed. So, this is how it is. The genes will do their duty. They will not mix up. So, according to his, during gamete formation, the factors of a character, which is also called as allele, which are present in the parent, they segregate from each other or they separate from each other, and in such a way that the gamete receives only one of the two factors. Here, only one factor is received and the dominant factor was received and the parent plant produced violet color. They did not mix up to form a pink color. So, that is what is meant according to the law of segregation. And homozygous parent, they produce similar gametes. If it is homozygous, see, this is what I said. If it is dominant homozygous, the parent will be violet itself. The plant progeny will be violet. Or if it is homozygous recessive progeny or the F1 generation will have white color flowers itself. And that is also one more point. Homozygous parent produces similar gametes and here also they will express similarly. And heterozygous parent produces two kinds of gametes wherein one will be dominant and the other will be recessive, having one allele with equal proportion. That is 1:1. One will be dominant and the other will be recessive. That is what is meant by law of segregation. So, these are the two laws of Mendel. So, one more, we said that we shall study about the non-Mendelian inheritance. That is a law wherein Mendelian inheritance is not followed here, or the two principles or laws of inheritance of Mendel is not followed here, and that particular is called as dominance, or it is called as incomplete dominance, the concept. So, before moving on to incomplete dominance, we should understand what the concept of dominance is. So, in heterozygotes, you know that there are two types of alleles being present. So, heterozygotes means they are dissimilar. So, one allele will be T and the other will be T. They are dissimilar. So, two alleles will be present wherein one allele will be dominant and the other will be recessive. So, there is one allele that is dominant and the other one is recessive, and the normal allele, that is that allele which is not unmodified or modified. So, that gene produces a normal enzyme and that is needed for the transformation of substrate. So, what they did was, in order to explain the concept of dominance, they took the example of an enzyme. So, they took the example of an enzyme. So, what an enzyme does? Enzyme is used to convert the substrate. Say, for example, in a human body, there are various enzymes that helps the DNA to produce different proteins. So protein is the substrate here. So they help to produce different proteins which will move to different parts of the body and perform its respective function. So that is why they use the example of an enzyme here. So similarly in a flower, not just in a human body, in the flower also the same genetic mechanism will take place. So here the normal allele, so those alleles which are unmodified, sometimes what happens is because of the external factors, a modification in the allele or the gene might occur. So in this case we are talking about the unmodified or the normal allele or the gene. So these normal genes or allele of a gene, they will produce a normal enzyme, and this particular enzyme is needed for the transformation of a substrate or converting a substrate into a protein or something, so that it can be utilized by the body in the case of plants and animals as well. So what they did was, so this is the concept of dominance. Like the allele, in the case of enzyme, they took the example of an enzyme wherein they said that if the enzyme, if there is a allele, that is a normal allele of a gene that is responsible for producing an enzyme, responsible for the modification or transformation of a substrate. Like normally, say for example, there is an enzyme which is having, there is a gene having two alleles and the alleles are normal. So that alleles, it will allow the enzyme to modify the substrate or transform the substrate, which is useful for the body. Say for example, now the allele gets modified. If the allele gets modified due to some external factors, what might happen? So if the allele is modified, then the normal, or then in that particular enzyme, the enzyme may become normal, or it may become less efficient, and the modified allele might produce the same phenotype like the unmodified allele. So what happens here is there won't be any change in the phenotype in spite of the modified allele. There won't be any change in the phenotype. So that is the codominance. So here, and also the modified allele may become dominant. The modified allele may become dominant, wherein it will suppress the unmodified allele to express. I told you, in a heterozygous species or a plant having a heterozygous allele in a gene, one allele will be dominant and the other will be recessive. Say for example here, the dominant allele is not modified, whereas the recessive allele is modified. So if one of the allele is modified, maybe it may function normally if it is a recessive allele, if it is a modified recessive allele, or if it is a modified dominant allele, then some changes may occur in the transformation of the production of the enzyme or the functioning of the enzyme. Therefore, it will not be able to properly transform the substrate. So that is what happens. And sometimes it may become dominant. One allele, if it is the unmodified or the modified allele, they may start to become dominant. So that also may occur, or sometimes it may become non-functional. The enzyme itself will become non-functional because the allele has been modified. So, and sometimes no enzyme at all will not be produced because of the modification. So all these may occur in the case of concept of dominance. So in the concept of dominance, so that is what is meant if one allele is dominant over the other. So if the modified allele is dominant over the unmodified allele, what all might occur? What will happen? The less efficient enzyme will be produced, and also the it may become dominant. The modified allele will start to become dominant, and the modified allele may become non-functional, and the modified allele may not at all produce an enzyme. So therefore the substrate, there is no conversion of the substrate. So this is all what happens in the concept of dominance. So here what happens here is, in the first and the second point, if you notice, the phenotype that will be produced. So if a allele is modified, and from that if a phenotype, if the parent allele is modified, and from that if a phenotype was produced, it will be dependent on the functioning of the unmodified allele. This is the beauty of a body, of a plant body or a human body. So a DNA tries to repair itself. To a number of times it tries to repair. So if there is any modification in the gene, nothing will happen to the plant, because the unmodified allele will suppress the modified allele. The modified allele will become recessive. That is what the concept of dominance means. So in the concept of dominance, according to point 1 and 2, what will happen is, if in point 1 and 2, the phenotype, it will be dependent on only the functioning. So what the phenotype of the progeny might look like, it will be dependent on only the functioning of the unmodified allele. What will happen is, the modified allele will get suppressed by the unmodified allele. That means the unmodified allele will be dominant and the modified allele will be recessive. So this is what is meant by. So the unmodified allele, it will become recessive. Here the unmodified allele will become recessive. So this is what happens here. So in 1 and 2, the phenotype will be mainly dependent on the functioning of the unmodified allele. Here the unmodified allele will become recessive. Therefore the factor will get expressed. So that is why here incomplete dominance will occur. So that is the concept of dominance. So moving on to non-Mendelian inheritance. So what is this non-Mendelian inheritance? It is nothing but incomplete dominance. So to understand incomplete dominance, I use the example of an enzyme, wherein the enzyme will have, you know that a gene will have two alleles, right? In that, one allele will be modified and the other will be unmodified. The modified allele will be it will produce the normal or the less efficient enzyme, and here the modified allele, even though the allele is modified, but in the phenotype it will not both the genes will be shared, both the modified and unmodified. So therefore it will not be a problem to the plant, and also it may become dominant and it may produce a non-functional enzyme, or it may not at all produce an enzyme. So this is what happens there. So let us study about the non-Mendelian inheritance. So non-Mendelian inheritance, why it is called non-Mendelian? Because it is not based on Mendel's law, that is Mendel's law of dominance and law of segregation. And here incomplete dominance is seen. So as I told you, here the genes will not get segregated. Both the genes may get expressed at the same time, resulting in the production of a different color of flower. Or both the genes, say for example, if there is a red color flower and a white color flower, both the genes will combine and it will produce a pink color flower. So that is what happens here. So to explain non-Mendelian principle, there is a best example where we shall select two plants, that is the snapdragon plant, which is also Anthurium species. It is a snapdragon plant. So a cross will be made between the snapdragon plant. So here we have R. So this is producing a red color flower and this one a white color flower. So here what happens here is, so when a cross is made, so here when a cross is made, you'll have RR, but according to the law of segregation, here the genes will not get expressed. So what you had studied previously in Mendelian inheritance, according to that, here it was supposed to be red flower, right? Because the allele or the gene responsible for the redness in the flower, it is being dominant here. But this will not occur here because it is the incomplete dominance. In incomplete dominance, what happens here is, instead of here either a red flower or a white flower being produced, the genes will not get segregated or they will not separate. They will blend with each other and a pink color flower will get produced. So pink will get produced. It will get modified. Now this is a modified flower. It is carrying a modified allele because there is no segregation of genes here. Therefore modification will occur and a pink color flower will be produced. So pink color flower will get produced. So now this pink we will cross and see what will happen. So what here in the F1 generation there was pink. All the flowers were pink in color. Pink flowers will be produced. So now a cross will be done between the F1 generation. So in the F2 generation, so when a cross is done between the parents of the F1 generation, we will see what will happen. Here it is red. Why it is red? Because there is no recessive gene involved here. Both are dominant, so it will not become the F2 progeny will not be pink. It will remain red itself. Here it will be pink because there is modification occurring here. That is, the genes are or the alleles are blending with each other. Here also it will be pink. What will happen here is there will be a change in the gene. Therefore, the unmodified allele will become recessive and the modified allele will become dominant and there is no segregation of gene. Therefore, a pink color flower will be formed. So here as usual it will be white color flower. So this is what happens. So in the F2, now we have after crossing we have how many red? 1, pink, 2 and white 1. So here what happens? We have 1 in the phenotypic ratio. There is 1 red flower, 1 red and 2 pink and 1 white. And in the genotypic ratio, 1 red, 2 pink and 1 white. There is no alteration in the phenotypic and the genotypic ratio. So this means that R, that is the dominant gene, was not completely dominant over R. According to 1 and 2, the phenotype will be dependent only on the function of the unmodified allele. Here the unmodified allele will become recessive. So that is what will happen. So though it is dominant R, it will become recessive. The unmodified allele will become recessive and it will not be completely dominant over R. Therefore, they will blend and become RR, which will be responsible for the production of a pink color flower. So this is what happens here. And P plants also showing incomplete dominance in the other traits as well. In the other traits also, again, if F3 generation is done, F4 generation, the same incomplete dominance can be seen. Not just in this. If we take, say in the case of incomplete dominance, if we select a yellow color, the pod of the seed of pea plant, if it is yellow and white, if we mix yellow and white, we might get a cream color. So it will change for other traits also. Or if we cross between a tall and a dwarf, in the case of incomplete dominance, what happens? If there is modification in the allele, what will happen? Maybe we will neither get a tall nor a dwarf plant. but we will also get a plant which is in between the height, neither tall nor dwarf. So for other traits also this is applicable. But here in this particular session I have taken the example of the snapdragon flower, wherein we took the red flower and the white flower, and what happened if the allele gets modified. So this is incomplete dominance, and because Mendel's law, that is law of segregation and law of dominance, does not apply here, it is also called as non-Mendelian inheritance. So incomplete dominance is also called as non-Mendelian inheritance. So according to the concept of dominance, what will happen? Always the dominant gene will suppress the recessive gene, right? But in this case, because of modification in the allele, what happens here is, though the dominant gene is there, it will not be able to completely dominate over the recessive gene or recessive allele. Therefore, their genes will not get segregated. They will blend together, or they will function together in a plant and result in the formation of a different color of flower, or that is in between colors. So this same thing will be carried for other traits or characters as well. So next, moving on to one more term called as codominance. So in that is non-Mendelian inheritance, along with incomplete dominance, there is one factor called as the codominance. So in codominance, it is the inheritance in which both the alleles of a gene are expressed in a hybrid. There, according to the law of Mendel, only one gene gets expressed, right? Only the dominant gene will be expressed and the recessive gene will be suppressed. But according to the non-Mendelian inheritance, here both the alleles will be expressed. So in codominance, it is the inheritance in which both the alleles of a gene will get expressed. The best example is ABO blood grouping in human. So in ABO blood groups, they are usually controlled by the gene called as I. So this is the gene that controls the ABO blood group. So previously you had studied gene R, which controlled the red color of a flower, gene small r, white color, or T, tallness, capital T, tallness, small t, short, dwarfness. So here it is the gene I that controls what blood group the progeny has to have based on the parent. So here, you know that in a in the blood there is the red blood cells. So the red blood cells, each cell is covered by a plasma membrane. So the plasma membrane of the red blood cell, they have sugar polymers. So these sugar polymers, they are how much amount of sugar polymer, how much amount it should be produced, all that it is controlled by the gene. So which gene controls that? It is the I gene. It is the I gene that controls all these things. It is controlled by the gene. So the gene I has three different alleles. It is called as IA, IB and small I. So the alleles IA and IB, they produce a slightly different form of a sugar, while so IA and IB produce, so what I told you, the RBC of the blood, that is the red blood cells, they are covered by a plasma membrane. On the plasma membrane, there is sugar being present. So sugar, the production of the sugar on the plasma membrane is controlled by the gene I. So it is controlled by the gene I. So in the gene I, there are three alleles, that is IA, IB and small I. So the alleles IA and IB, they produce a different form of sugar, while the allele I does not produce any sugar. Why? Because small I, it is recessive. Capital I, it is dominant. So based on the dominant I and the recessive small I, it will decide in that red blood cell, in the plasma membrane of the red blood cell, whether the sugar should be produced or not. So based on whether the sugar is produced or not, the blood group of the progeny will depend. So here, when IA and IB are present together, they both express their own types of sugar, and this is called as codominance. Both of them will express their own kind of sugar. It is not that because here both are dominant. They will express both the kinds of sugar. It is not that IA will suppress IB and it will get expressed. No, it is not like that. They will act mutually. That is, they will produce separate set of their own sugars. Based on that, the blood group will be decided. So we can see here. So we have the blood group IA, IB or I. So these are the alleles that are usually present in the male or the female. In the female, there is the IA, IB and I, and in the male also we have the IA, IB and I. This will not produce, I will not produce sugar. This particular allele will not produce any sugar on the RBC, and these two will produce their own set of sugar or their own respective sugars on the plasma membrane of the red blood cells. So what happens here is, if the female parent has the allele IA, and if the male parent has the allele IA, then the progeny will have the blood group IAA, which means it will have A blood group. The progeny's blood group will be A. It will be A. If, because the sugar that is expressing there on the RBC, plasma membrane of the RBC is A. It is dominant. That is why the blood group will be A. And if the female parent has a blood group IB as the allele IB, and the male parent has IA, then both will get expressed separately. Why? Because I told you, according to here, if IA and IB are present together, they both will express their own types of sugar. That is why they get expressed separately. They get expressed, and the blood group will be AB. You might have seen the blood groups AB positive, right? AB. And if the female parent has a recessive allele and a dominant allele IA, then it will be IAI. Again here the blood group will be A. It will produce A blood group. And if the male parent has IB allele and IA allele is in the female parent, then the progeny will again produce here AB blood group. The progeny will have a blood group AB. And if the male parent has allele IB and the female parent has also allele IB, then the blood group will be B, because here they are getting expressed B. And again here, if it is IB in the male and I in the female parent, then the progeny or the offspring or the baby will have the blood group B expressed. Because this is recessive, it will not get expressed. I is recessive, it will not get expressed. Only B, blood group will be B therefore. And if the male parent has a recessive allele and a dominant allele IA, then it will be produce a blood group A. So different combinations. So if the male parent has the blood group recessive I and dominant IB in the female parent, then again here be it a male parent or the female parent, no problem. It always depends on how the recessive allele is being present. Depends on mainly on the recessive allele. So here the blood group will be B. So here again, if the male parent has a recessive allele I, and again the female will also have a recessive I. Here you should remember, no sugars will be produced on the plasma membrane of the red blood cells. Therefore, the blood group will be O. And majority of the people, they have this O positive or O negative blood group. The progeny or the baby will have O blood group. So you can calculate by asking your parents what is your blood group. You can calculate like what your blood group is. Then you'll easily find out. So you can do this task at home, and then you can easily find out. So what are the different blood groups that are present? A, AB, B and O. So this is about codominance. Both the alleles are being expressed here. Only the recessive allele will not get expressed. But if both the alleles are dominant, both will get expressed separately. That is why it is called as codominance. They are cooperating with each other, codominance. So next there is one more factor called as pleiotropy. So in pleiotropy, only one gene will produce more than one effect. That is, I told you, if the red flower or is there and the white flower is there, only one gene will be expressed, right? Either the red or the white, depending on whether they are heterozygous, homozygous, dominant or recessive. But in this case, in the case of pleiotropy, what happens is one single gene will produce more than one kind of an effect. Say for example, we shall take the example of the synthesis of starch in pea seeds. So pea, it is a dicotyledon. So in the pea plant, there is the endosperm that is being present. What is the function of the endosperm? To produce starch. So the pea seed has starch. So we shall see the synthesis of starch in pea seeds. And one more example is there, sickle cell anemia. It is a blood related disease, sickle cell anemia. So here in this session, we shall discuss keeping the starch in pea seeds as an example. So the starch synthesis in pea plant, what happens is, that is the synthesis of the starch in the pea seeds, it is usually controlled by one gene which has two alleles, B and B. So both are dominant here, B and B. It is controlled by two alleles, dominant B and dominant B. So starch is synthesized by both BB, because they are dominant, more amount of starch will be synthesized because both the alleles are dominant there. So by the dominant homozygotes, and therefore large starch grains will be produced, while the BB homozygotes, which have lesser efficiency, here it is not that, I am talking about pleiotropy. One gene will express both the characters, that is more than one character a single gene will produce. Here it is not that BB will get expressed and this will get suppressed completely. No. Here both will express separately, wherein if the alleles, both the alleles are dominant, then the large starch granules will be produced. Large starch granules. And if it is recessive, again it will get expressed. It is not that starch will not at all be produced. Starch will be produced, but small granule starch will be produced. Small granule starch. So this is about pleiotropy. So what happens? It has two alleles, B and B. If both the alleles are effectively expressed, then the starch, large starch grains will be produced in the pea seeds. And if it is, if B is expressed, that is the recessive BB is expressed, then also the starch will be produced. It is not that the completely starch will not be produced. Starch will be produced, but they will have less amount of starch will be produced. The starch, the granules of the starch will be smaller in size, and all that will occur. So after maturation of the seeds, the BB seeds are round and the BB seeds will be wrinkled. This is what happens here. Why is the BB seed round? Because it is filled with starch. That is why it is round. Because it is getting proper nutrition, right? Why is the BB small? That is this seeds having the recessive allele, they will be wrinkled. Why? Because starch is less produced. They will have less nutrition. That is nothing but pleiotropy. So in pleiotropy, the heterozygotes will usually produce the round seeds, and so what happens here is B will be the dominant allele, and but the starch grain produced are of intermediate size. That is like if BB, if you mix, that is BB, then what amount of starch will be produced? They will be intermediate. Again, what happens here is, so if you mix BB, again here also starch will be produced, but it will neither have larger starch grains nor smaller starch grains. The size of the starch grains in the pea seed will be somewhere in between. So that is what is meant here. So heterozygotes, they produce round seeds, and so the seeds will be round, but the amount of starch that is produced will be different. So heterozygotes, they will produce round seeds, and so why? Because B is the dominant allele, but when it comes to the starch grain that is produced, they will be of intermediate size. So I hope you have understood here, because it is having the B allele, the seed will be round itself. In the case of the gene being BB. But because there is one more recessive gene being present, the starch grains will be of intermediate size, that is intermediate, that is it will neither be large nor it will be small, like how we had seen in BB and BB. Neither it will be large as in the that is the seeds, P seeds having the homozygous dominant BB trait and the homozygous recessive BB character, but it will blend with each other and the seed will be round as such because B is dominant. But in spite of having a different allele here, both the allele being heterozygous, in spite of that, there will be the starch grains will be intermediate. That is, the size will be neither large nor small, it will be intermediate. So therefore, dominance is not a autonomous feature here, like in how in Mendel's experiments we had seen. If it is dominant, the entire recessive gene will be suppressed, right? But in this case, both the importance will be given to the dominant as well as the recessive. Both will get expressed. That is what it is mentioned here. So that is what pleiotropy means. So in pleiotropy, what happens here is a single gene will produce more than one kind of effect. It is not that one particular gene should suppress the other. Both the genes will get expressed on their own. So that is why different sets of characters in a progeny can be found in the case of pleiotropy. So this was about this particular session wherein we studied about the non-Mendelian inheritance. So in the next coming session, we shall see about the inheritance of two genes. So far in the previous session and this session, we had studied only the monohybrid cross, that is wherein the plant only one trait or one gene will be considered in each plant. So in the next session, we shall see what will happen to the progeny if two traits of a parent are considered, that is inheritance of two genes in a progeny we will study. And also we will study about the law of independent assortment. So I hope you have understood this session very well. So we shall meet in the next coming sessions. Thank you.